Good afternoon, everyone. Thank you so much for joining the NOAA Central Library and our presentations today from the 2020 Knaus Fellows. For your information, I am Katie Rowley. I am a librarian and I will be hosting this webinar. If you have any technical issues, I recommend you logging out and logging back in. And if you have questions for the speakers today, please place those in the question panel. We will try to get to all of your questions. If we don't, they will be sent to the presenters after the webinar. And with that, I'm going to turn it over to Caitlin Louder to introduce our speakers.
Hello, our first speaker this morning is Anne-Christine Zinkann. Anne defended her PhD in marine biology at the University of Alaska Fairbanks, focusing on organic matter source contribution to lower trophic level food webs, with a specific interest in terrestrial and bacterial organic matter. Her research focused on determining how different organic matter sources are represented in marine sediments and utilized by benthic invertebrates to then update a current Chukchi Sea ecosystem model. She is currently a Knaus Fellow in the Global Ocean Monitoring and Observing Program. Welcome, Anne.
Hey, thank you for the introduction, and I'll get right into it. The title of my dissertation is Organic Matter Sources on the Chukchi Sea Shelf in a Changing Arctic. And at this point, I would like to thank my committee, Katrin Eichen, Matthew Wooler, Seth Danielson, Mary Beth Lee, and Georgina Gibson, who are all professors or associates at the University of Alaska Fairbanks. And I will go over the 3 chapters of my dissertation today to give you an idea of what I was doing for the last 5 years. So let's get started. Oops. There we go. So the Arctic Ocean is experiencing climate-driven increases in temperatures at rapid rates. And Arctic shelf systems are particularly vulnerable as they're experiencing warming 3 times the global average. And current estimations predict that the Arctic Ocean is to be ice-free in the summer as early as 2040. And among all Arctic shelves, the Chukchi Sea shows one of the fastest declines in sea ice cover, which you can see on this panel. The Chukchi Sea is a highly productive shelf of the Arctic, located between Siberia and Alaska. And the top panel shows the average sea ice cover for the summer months between 1979 and 88, with the dark blue colors being open water and the light blue to white colors indicating sea ice cover, while the bottom panel represents the average sea ice cover from 2007 to 2011. And these changes in sea ice cover can have a variety of consequences, and one of them are the strength of pelagic-benthic coupling, which I will go into in the next slide. This is a very simple depiction of an Arctic food web with the benthos at the bottom, which just represents the seafloor, the pelagic realm, which is the water column, the ice cover or sea ice, and the ice edge bloom consisting of sea ice, algae, and phytoplankton. And in strong ice conditions, this favors an early primary production bloom when zooplankton communities are not quite developed yet, and therefore grazing rates are relatively low, causing most of their production to sink to the water column to the seafloor, and the system being in a tight pelagic benthic coupling. With earlier sea ice retreat, this allows for a stronger wind mixing, resulting in a later formation of the pycnocline, causing a delayed phytoplankton bloom that is then largely consumed by the zooplankton population, switching the system to from a tight pelagic benthic coupling to a weak pelagic benthic coupling, meaning that less of the pelagic production reaches the seafloor and can sustain the benthic biomass. And until recently, in Arctic food web studies, they mainly concentrated on sea ice, algae, and pelagic production as the main food source for these benthic food webs. But recently, other organic matter sources have been identified to contribute to the benthic invertebrate diets, and these include terrestrial matter and water and sediment bacteria. But why do we care so much? So for phytoplankton, sea ice modulates the amount of light that is available for photosynthesis. And phytoplankton communities in the Arctic have historically been dominated by large diatoms that are very heavy and sink very fast out of the water column. And although increased light availability in a longer growing season allows for increased Arctic phytoplankton production to occur, we also have been observing an increased influx of boreal species that are much smaller, and a change in water temperature, nutrient availability, and especially the freshening of the water seems to favor these smaller phytoplankton cells, which are much lighter and sink much slower. In addition to that, we have terrestrial matter. And although the Arctic Ocean is one of the smallest oceans, it receives a disproportionately large amount of river discharge. And since the 1970s, river discharge onto the shelves in the Arctic have increased by about 2.6% per decade, as well as coastal erosion. And with that, the amount of terrestrial matter that is carried onto the shelves and into the Arctic. And the majority of river discharge on the Chukchi Sea shelf comes from the Yukon River. And although the Yukon River has only shown small changes in its discharge over the recent decades, in combination with an increased throughput of Pacific water through the Bering Strait, these small changes can have high impacts of this contribution of terrestrial matter to the benthic, the benthic realm. And last but not least, we have bacteria. And besides being beautiful art projects, they're mostly consumed indirectly by benthic organisms by being attached to sediment and algal particles. And they're an important food source as they contribute nutritional value to the detrital food that they're mainly feeding on. Bacteria are, of course, important in breaking down organic matter and are critical in recycling nutrients back to primer producers to fuel new production, and at the same time supply a food source in terms of bacterial biomass. And rising ocean temperatures are predicted to cause an increase in bacterial activity, and therefore the potential for this organic matter source to become more important to benthic invertebrates in the future with rising ocean temperatures. So as these three organic matter sources settle down to the sea floor or to the benthos, it supplies organic matter to these communities that they're directly feeding on. And in the Chukchi Seas particularly, the Chukchi Sea ecosystem is dominated by benthic biomass, and the majority of the biomass of this ecosystem lies in the benthic realm. They also play a vital role in influencing detrital degradation of organic matter and sequestration of this matter into sediments, and they're an essential ecosystem component and serve as an important energy link between settling organic matter and higher trophic levels, including fish, birds, and marine mammals that are of course important for fisheries and rural communities. And these are just some examples of the benthic predators that are important for the shelf system. And just to give you an idea, gray whales annually migrate to feeding grounds in the Chukchi and Bering Sea, where they scoop up sediment with the side of their mouth and then filter out small little crustaceans that they're then feeding on. But these feeding migrations and areas that they're occurring is shifting at the moment. So this brings me back to the general Arctic food web depiction that I introduced in the beginning. And we know that all of these three sources are currently changing, but what we don't know is to what proportions are they actually present in that system and to what proportions are they being utilized by organisms. And this leads me to my research questions. And they were threefold. One, what organic matter sources are in marine sediments? What organic matter sources are actually being utilized by the benthic invertebrates? And then what are the potential implications on the ecosystem with changing organic matter supply? And this directly relates to the NOAA Arctic Vision and Strategy in strengthening foundational science to understand and detect Arctic climate and ecosystem changes. So to answer these questions, I went out into the Chukchi Sea as part of the Arctic Marine Biodiversity Observing Network cruise in 2015, where I sampled sediment samples across the shelf, and you can see a little map down here, and invertebrate tissue samples, and brought them back to the school where I then analyzed them for stable isotopes. And I will very shortly and briefly go over this. So isotopes are atoms of the same element with a different mass. And the stable just refers to that they don't decay over time. In major taxonomic groups, for example, there are two examples right here, have different ways of acquiring and fixing carbon that leads to different carbon isotope ratios, whereas we like to call them delta 13C values, which is the little sign down here. And as consumers feed on these organic matter sources, they conserve this delta 13C value within that tissue, which allows scientists to then use it as a proxy of tracking the utilization of these different organic matter sources. And you can kind of think of it as a fingerprint. So the euphosit feeds on, for example, phytoplankton, and then has a fingerprint of the phytoplankton in its tissue. And we're looking at the fingerprint to then backtrack where it came from. And usually what we use is bulk stable isotope analysis, which looks at the overall total organic matter content with all compounds in an organism's tissue to give us an idea of what they're eating. However, one of the drawbacks of bulk stable isotope analysis is that we're only looking at one point on this fingerprint. And we know that environmental conditions can influence carbon isotope ratios. And then you can think of it kind of, it smudges the fingerprint a little bit, and then it makes it harder to distinguish among various organic matter sources. So I decided to utilize compound-specific stable isotope analysis, which targets not the bulk tissue, but specific compounds of the tissue. And I used amino acids. There are 20 amino acids present in all life forms, and about half of them are considered essential. So they can only be synthesized by bacteria, fungi, and photoautotrophs. And they're essential because they have to be taken up by an organism's diet and cannot be synthesized by the organism themselves. And so if you think about the fingerprint approach again, we're looking at multiple essential amino acids. So we have multiple dots on that fingerprint that then help us to backtrace where this fingerprint originated from. And we use mathematical equations to then calculate proportional contributions of these different organic matter sources that contributed to the organism's fingerprint. And to dive in right into some data, my first chapter was looking at organic matter sources in sediments. And to dive right in, here is a map of the Chukchi Sea shelf. And each pie chart represents one station that I sampled and represents the first 2 centimeters of the sediment. And the colors will remain the same from now on, with yellow being bacteria, light green being phytoplankton, and dark green being terrestrial matter. I did not find any significant differences across the shelf. I also checked for potential correlations with any of these environmental variables and did not find any strong correlations. The only thing that stood out were these 2 southernmost stations that had very high contributions of terrestrial organic matter. When I then looked at the depth of the sediment, the first 5 centimeters that I chose to look at, you have sediment depth on the x-axis and proportional contribution on the y. And each of these panels represents an organic matter source. What I want you to take away from this is that I did not find any significant differences with depth except for phytoplankton, which if you look at the letters above the bars here, those indicate significant differences. Phytoplankton showed a significant decrease in the proportional contribution in sediments. And what I also would like you to note is that terrestrial matter was present in very high quantities in all depth strata. So in conclusion for sediments, I did not find a spatial pattern, which I allocated to be partly due to that the stations were located in the same water mass. The Chukchi Sea has multiple water masses, and we did check where our sampling stations were located, and they were all the same water mass. There was mostly no significant difference with depth, which indicates a well-mixed top 5 centimeter layer, which was likely due to the bioturbation activities by benthic invertebrates. And the high amounts of terrestrial matter are likely due to the river discharge from the Yukon River, coastal erosion, and the long degradation times that it takes for terrestrial matter to be degraded. So what I wanted to know next is, what do invertebrates actually eat from what we just established is the baseline? For that, I pulled them into different feeding types, and I will shortly go over them and what that means. So we have 3 major feeding types. We have deposit feeders that are separated into surface deposit feeders that feed on material deposited on the sediment surface. And we have subsurface deposit feeders that feed on sediments a little bit deeper. We have suspension feeders that capture settling and resuspended particles, and predators and scavengers that hunt their prey. And this feeding activity usually accounts for them utilizing different organic matter sources to a different degree. And what I also want to note real fast is that when I was sampling for these organisms, I tried to get multiple species that represented these feeding types. So that's what these N's mean in the behind the different feeding types. So diving into the data, here each panel represents a feeding type. You have all organic matter sources on the x-axis and proportional contribution on the y. Colors again are the same, and error bars are standard deviation. What I want you to take away from this is that I did not find any significant differences within the feeding types, except for slight trends towards higher terrestrial matter contribution to both suspension feeders and surface deposit feeders, and a higher contribution or trend to higher contributions of bacterial organic matter in subsurface deposit feeders and predators and scavengers. And the variability here was really, really high. So what I wanted to know next is this variability does it stem from me pooling together multiple species? And this is what you see here. I have each feeding type in a panel, and each pie chart represents one species that represents this feeding type. And what I want you to take away from this is that the variability within and among feeding types is very, very high. So for example, if you look at the suspension feeder, the bryozoan Alcyonidium, the contribution of terrestrial organic matter is about 70%, while the bivalve Cirripes only has just about 25. And this pattern is present in most other feeding types as well. So if you look at predators and scavengers over here, you have the gastrobot Buccinum, which has over 75% terrestrial matter contribution to its diet, while the shrimp Eualos only has about 10. So we have a very, very high variability, even within the same feeding type. So in conclusion for the invertebrates is we have high contributions of terrestrial matter to all feeding types, which is likely due to the high proportions that are present in all sediments. We have no significant differences in organic matter proportions among the different feeding types, which is likely due to the lack of a strong pattern across the shelf. And there's a high variability within and among feeding types, which I attributed to be due to additional characteristics that I did not account for in this study, including mobility, selectivity of feeding, and assimilation efficiency of the gut. But except for giving us like a baseline of what the system looks like right now, what I wanted to do is have a model and update a model looking at future impacts and accounting for these changes that I introduced in the beginning. And I utilized an ecosystem model called EcoPath and EcoSim. EcoPath allows me to build a mass balance model, and EcoSim allows me to force this mass balance model into a certain direction to see how the ecosystem reacts to that. And ecosystem models are really useful, especially when you want to conduct an experiment that is just not feasible to do. So introducing the Whitehouse and Iden 16 model, which is the current Chukchi Sea model. They build a mass balance model representing all the major groups: marine mammals, birds, fish, pelagic and benthic invertebrates, and then some of the detrital groups and phytoplankton. And this is my modification. So I introduced a terrestrial component based on my findings from my first 2 chapters. It then allowed me to update or revise the phytoplankton production values to recent findings, and I introduced benthic invertebrate diets that focused on these 3 carbon source or organic matter sources that I introduced. And this is how it all looks like. And I know that's very confusing or overwhelming, but I'll explain it real fast. So you have trophic levels here on the y-axis, and each dot represents a group or a specific species. The colors correspond to the bigger groups. So orange, for example, here is benthic invertebrates, yellow are being fish, dark blue are marine mammals, and so on and so forth. And the size of the dot indicates the biomass. So it's very evident here that the majority of the biomass lies in the benthic invertebrate pool of living organisms. And each line, each of these gray lines indicates a feeding interaction. So For example, this gray line goes to jellyfish, so we know that jellyfish feeds directly on phytoplankton and so on and so forth. And this is the mass balance model that I updated, which just mass balance just means that all the energy that goes in goes out. What I did next is utilize EcoSim, which is the simulation component. And as I introduced before, I had 4 simulations. The first simulation forced the system to have an increased phytoplankton biomass, which is predicted to increase by about 10% over the next 35 years. Then I weakened the pelagic benthic coupling in my second simulation. My third simulation accounted for an increased terrestrial matter inflow through river discharge and coastal erosion. And my fourth simulation accounted for an increase in bacterial production. And I will shortly go over a couple examples so you know how this all looks like. So you have on the x-axis the time scale, and the y-axis represents a change in relative biomass. This little gray dotted line is 0% change, and each of these colored lines is one of those groups that I introduced before, one of those little dots. And the color coding is just based on the patterns that I found, mainly on the feeding activities. So this is the first simulation where I increased phytoplankton biomass, where we see a positive response, more relative biomass in benthic invertebrates. When I then switch the pelagic benthic coupling to a very weak system, we see a decrease in benthic invertebrate biomass with less response in these dark lines, which represents organisms that majorly or predominantly feed on terrestrial food sources. When I then, in addition, increase terrestrial organic matter inflow, we see partially a recovery of some of these benthic invertebrates, but the ones in these light lines that are organisms that majorly feed on phytoplankton are still not recovered. But when I then also in addition increased bacterial production, we see a recovery in all of these benthic invertebrate groups. And then I did this for all the major groups, and I'm only going to show one other just to give you an idea of how this looks like. These are marine mammals with the dark blue lines being pelagic feeding and the light blue lines being benthic feeding. And we see a similar response as we see in the benthic invertebrates, and the light blue line down here and up here, the one with the strongest responses, those are actually gray whales that are responding to the changes in the crustacean biomass in benthic invertebrates. So as a conclusion for my modeling chapter, I incorporated terrestrial matter source into the mass balance model, which allowed me to reduce and revise phytoplankton production numbers. And it represents now all utilized organic matter sources in the system. And a weakening in pelagic benthic coupling was able to be compensated by an increased inflow of terrestrial matter and microbial production. However, please take this with a grain of salt, because this is all based on feeding interactions only. It does not, for example, take into account any metabolic demand changes by benthic invertebrates or higher trophic levels, changes in community composition, or interactions with other things like the association of cod juveniles with sea ice cover. And it does not take into account any nutritional value. So whether terrestrial matter is less nutritionally valuable than phytoplankton. So for overall outcomes, my dissertation showed that terrestrial organic matter is a significant organic matter source in both Chukchi Sea sediments and food webs. And future shifts in organic matter source composition to the sediment both have implications across the entire ecosystem, and a diversification of organic matter sources at the base of the food web could potentially mitigate some of the negative impacts that we're expecting with a change in or a weakening in this pelagic benthic coupling. And further studies would be great if they could concentrate on nutritional values in terrestrial and bacterial organic matter, as we do not know at this point what those are, and quantify better the influx of terrestrial matter and which one is usable, and quantify bacterial production under various environmental conditions. And with that, I'd like to acknowledge all my committee, my funding sources, which are on the right, all the crew from the Norseman 2 and the Skoliak, everyone from the Alaska Stabilized Facility, Andy Whitehouse, Diane O'Brien, and Billy Christensen, and Brian Terrace, who were super helpful with the model and the stable isotope parts, my lab, and my parents and my friends. And with that, I take any questions.
Thank you so much, Anne. I'm not seeing any questions pop up yet. Oh, and I am corrected. We have a couple. So the first one comes from Christine Bassett. How long-lived are the calcium carbonate precipitating invertebrates?
Can you repeat that?
Yes, sorry. How long-lived are the calcium carbonate precipitating invertebrates?
That's a really good question. It depends on which ones you look at particularly, but they're relatively long-lived due to them being in the Arctic, which is cold and they have very slow growth rates. So I think if I remember correctly, for some of the crab species, we're looking at probably about, I don't know, 6 to 10 years, I think if I remember correctly, but I didn't specifically look at that. But that's a really good question.
Great, thank you. Our second question comes from Eric Weisberger. Do you think accounts for the higher terrestrial values at those two southern sites? What do you think of the accounts?
So I was digging a little bit into literature, and it looks like that area is, so as the Pacific waters push through the Bering Sea, it gets pushed through at very high rates and high speed, and that area seems to be where water masses slow down. So we were thinking that it could potentially be just a spot where we have a lot of precipitation that settles out to the seafloor as the water slows down. That was one of our thoughts on that.
Great, we have a couple more questions coming in. Which benthic suspension feeders did you look at?
I looked at the Bryozone, Alcyonidium, and Seripes, which is a bivalve. We tried to get a couple more, but yeah, we tried to choose the ones that have the most representation across the shelf. So.
Great, thank you. Next question, how might you account for or measure seasonal variability in terrestrial contributions?
That's a very good question. I'm not sure I have an answer for that. Well, at the moment, what I looked at when I was trying to get an idea how much terrestrial matter is being swept into the Chukchi Sea, I was looking at, there's an online portal and the name escapes me right now, but they're measuring the river discharge and they're basing it on particulate organic matter so they're tracking that over time. There's a couple other methods, and I can look them up again, that look at the usable part of terrestrial organic matter. So which part of the organic matter that's actually being swept in is usable for benthic invertebrates. But yeah, I didn't dive into that too deep, but I can definitely look that up again and send some papers.
Great, thank you. Our next question, did you characterize the terrestrial OM? If yes, how did you do it?
I'm not sure if I understand the question, but yes, I guess what you're trying to ask is how I determined that it was from terrestrial matter. So I'm just going to go with that. So there's some studies by Larson et al. That looked at specifically Arctic terrestrial plants, and they determined the essential amino acid stable isotope values for all of those, and then compared those to different phytoplankton species, different red algae, brown algae, and bacterial species, and looked at whether the average of those is different enough for them to be able to be differentiated. So using the essential amino acid pattern of those, that's how we distinguish them. And I used we had another student in our lab that was looking at contributing a couple more samples for terrestrial plants to that database so we can get a little bit better of an idea of what that baseline looks like. So yeah, so that's what I used. It's basically tracing back the fingerprint that was established by another scientist a couple of years ago. And I'm happy to expand a little bit more on how this all works.
Thank you so much, Anne. We've reached time for your presentation. If everyone will give us a minute, we are going to switch over to our next presenter.